Welcome back, guys. So this video will going to be the second video of our major histocompatibility complex playlist. So I have already uploaded a video on MHC. So if you have not watched it yet, and in case if you do not have any knowledge on MHC, so please go and watch the first video, then come back here, then it will be helpful for you. Okay, so let's start. First of all, MHC genes. We have talked about three important characteristics of MHC gene in our previous video, and they are: MHC genes are polymorphic. That means multiple alleles are present for each MHC gene, right? MHC genes are codominantly expressed, right? And they are inherited together as a unit, right? And this each unit we have called as haplotype, right? Now today, as a continuation of that part, I want to told you that there are various MHC genes present. Those are polygenes or polygenic. Okay, and I hope you know about the polygenic trait. That is, our skin. Our skin color is controlled by many genes. Okay, similarly, there are various type of functions performed by MHC molecules, that is controlled by many genes. Okay, that is why MHC genes are also polygenic. Okay, that means genes having similar function. Okay, so we have learned MHC genes are polymorphic. We have learned MHC genes are codominantly expressed. We have learned MHC gene inherited together as a unit, and we have called those unit as a haplotype. Then we have talked about the concept of polygene, right? That is, MHC genes are polygenic. Okay, now if we want to classify MHC gene, so we can classify them as class 1 MHC gene and class 2 MHC gene and also class 3 MHC gene. Okay, I want to told you that among them first 2 is very important and class 3 is not too much important, although we will going to make separate video for class 1, class 2 and class 3. Okay, now if we talk about class 1 MHC gene, so this class 1 MHC gene will going to produce MHC protein, obviously, and those MHC protein will be known as class 1 MHC protein. Okay, now among this class 1 MHC protein there are two types. Okay, one is known as classical MHC gene and another one is known as non-classical MHC gene. Okay, so what are classical MHC gene? Classical MHC gene are those who have the ability to play a important function in antigen presentation. Okay, on the other hand, non-classical genes are those who have the ability to play, that means who do not play any kind of role in antigen presentation. Okay, so classical genes will produce protein that will involved in antigen presentation. Non-classical gene will not going to do anything that will going to play a role in antigen presentation. Okay, so that is the idea of classical MHC gene and non-classical MHC gene. You will also get the idea of classical MHC gene and non-classical MHC gene in case of class 2 also. Okay, and there the meaning is also the same. That means classical MHC gene will play an important role in antigen presentation, but non-classical gene will going to do some other jobs but not antigen presentation. Okay, now what is the meaning of this antigen presentation? Now, as I have not uploaded any video on the basics of immunology, so I want to mention this antigen presentation thing in a very simple way. That is, suppose let's say there is a very well known gang in your society. Okay, and anyone know do not know, or anyone do not have any kind of idea about the members of that gang. Okay, now let's say during a operation of those robbers, that means here I am not using the word operation as a term of good operation. Okay, here I am using operation meaning that those gang are going to do some mischiefs. Okay, so during this operation, let's say some victims recognizes or watches the face of the robbers or some members of that gang. Okay, now if that victim will survive, then after that he will go to the police station, or he will ask the police officers that, sir, I have watched or I have recognized one of those gang members. Okay, so what now that police will do? Police will bring a good sketchers, right, who will going to sketch the portrait of that gang member. Okay, now that portrait will be sent to every police station. Okay, and so that every police station can recognize that gang, and whenever they will find him, he will be imprisoned, right? So similar case happens in case of our immune system. Okay, and here the policeman is the MHC molecule. Okay, MHC will going to process the antigen, and here processing means MHC will going to cut the antigen. Okay, and take a special part of antigen molecule. Okay, and present them. Okay, and as a result those molecules can be recognized by other type of immune cells. Okay, that is other police stations, and now depending upon their features they will work. Okay, that is how MHC works, and that is the concept of antigen presentation. Okay, when we will going to talk about T cell, B cell, when we will going to talk about macrophages, then you will understand it more clearly. Okay, now so remember, class 1 MHC molecule will going to present antigen to cytotoxic T cell. Okay, and we also can call TC cell as CD8 plus T cell. Okay, that means in cytotoxic T cell, CD8 molecule will be present in its surface. Okay, similarly class 2 MHC molecule will going to present antigen to helper T cell. Okay, so now let's talk about class 1 MHC gene. So I have told you that class 1 MHC gene can be divided into classical class 1 MHC gene, non-classical class 1 MHC gene. Okay, so now classical class 1 MHC gene can be or is consist of 3 genes: HLA-A, HLA-B, HLA-C. So I hope you know about the full form of HLA, that is human leukocyte antigen. So these 3 genes together will going to produce 3 class 1 MHC molecule. Okay, and non-classical class 1 MHC gene will not going to take part in antigen presentation. We are not bothered about it. Okay, so that is the first photo. Now let's move into the second. So class 1 MHC protein is coming from where? Class 1 MHC proteins are coming from class 1 MHC gene, right? That is very clear. Okay, now these class 1 MHC proteins, remember, are glycoproteins. Okay, and this class 1 MHC molecule are nearly expressed on all nucleated cell. Okay, we have talked about nucleated and anucleated cell in our complement systems too, right? I hope you have watched it, right? So now class 1 MHC molecule are expressed in nucleated cell but not in anucleated cell. That means you will not get MHC molecule on the surface of RBC. Okay, so here are some important MCQ that high level of class 1 MHC molecule are expressed by lymphocyte. Neuron, sperm cell lack class 1 MHC molecule. fibroblast and liver hepatocyte neural cell express very low level of class 1 MHC molecule. Okay, now so let's move on to the structure of class 1 MHC molecule. So this is the structure of class 1 MHC molecule. Okay, it consists of two peptides. Okay, broadly, first of all, it consists of one alpha chain. This part is alpha chain and a beta 2 microglobulin. Okay, so two parts are, one is alpha, another one is beta. Okay, now we can divide alpha into three domains: alpha 1, alpha 2, alpha 3. So this part is alpha 1, this part is alpha 2, this part is alpha 3. Okay, and then this is beta. Okay, so now this alpha part is about 45 or 43 kilodalton. Okay, and they are transmembrane glycoprotein, remember it. And beta is 12 kilodalton. Okay, and there is no transmembrane region present on beta. Okay, as you can easily see it, right, that beta is, there is no transmembrane region in case of beta, but in case of alpha there is transmembrane regions, right? Now this beta 2, now what do you think, that which one is more important, class 1 alpha chain or beta? Try to answer it. So let me introduce that class beta 2 microglobulin is more important. Okay, because if beta 2 microglobulin is not produced, class 1 alpha cannot be expressed on cell membrane. Remember it very carefully. And this is a kind of disease, and the cell having this kind of disease is known as Dowdy tumor cell. We will going to talk about this Dowdy tumor cell later, but the idea is same, that in case of Dowdy tumor cell cannot produce beta 2 microglobulin, and as a result class 1 alpha chain, despite it is produced, but it cannot be expressed on the cell membrane. Clear? Now, so remember in the first video I have told you that most of the MHC gene are polymorphic, right? That means they are having many alleles, but some of the MHC genes are conserved. Okay, so remember the genes which produce alpha 3 part are conserved. Okay, and there is a valid reason for being conserved. I will going to talk about this. And alpha 1 and alpha 2 domain are coming from genes which are highly polymorphic. Okay, now another important thing, that alpha 1 and alpha 2 domain together create a groove known as peptide binding groove. Okay, this peptide binding groove will going to present the antigen. Okay, so that means whatever the MHC class 1 molecule will made, it will present it with the help of this peptide binding groove. Okay, and remember this peptide binding groove can take amino acid of 8 to 10 amino acid containing peptide. Okay, that means any antigen, no matter how long it is, it should be cut in 8 to 10 amino acid long peptide. Okay, unless it cannot be expressed by class 1 MHC molecule. Okay, clear? Now, so now how they will present? So remember there is a special type of sequence present on alpha 3 that will going to recognize CD8 plus protein, which is present on what? TC cell, right? And that is why I have told you that alpha 3 is conserved, because try to feel it, that alpha 3, if alpha 3 was not conserved, CD8 plus cell cannot interact with itself, right? So how important alpha 3 is, and that is the reason why it is conserved. Okay, that means after it is produced by MHC class 1 molecule, it will be transferred to CD8, or it will be presented to CD8 plus T cell with the help of alpha 3 also. Okay, now, so that is the overall idea. So here what I have told you, 3 external domain, alpha 1, alpha 2, alpha 3, transmembrane domain of about 25 it will be 25, as I am a Bengali student, sorry for this, it will be 25 hydrophobic amino acid followed by a short stretch of charged hydrophilic amino acid. Now cytoplasmic anchor segment of 30 amino acid. Okay, so that is all about the alpha chain, and beta chain does not contain transmembrane region, and it is non-covalently bind to MHC class 1 alpha chain. So another important information that this beta 2, this alpha chain, and the peptide molecule that will going to bind over here, all are joined together, or all are connected together via non-covalent interaction. That means all type of interaction here are non-covalent. Okay, remember it. So now the last one. So that is, I have already discussed with you. This part is about the peptide binding group, that alpha 1 alpha 2 domain interact to form a fluor of 8 antiparallel beta strand. Please remember it, that if it is class 1, let's say if it is alpha 1, if it is alpha 2, so this part is 8 antiparallel beta strand, and these two sides are alpha helix. Okay, that means alpha helix and 8 antiparallel beta strand together forms the groove or cleft. Okay, where 8 to 10 amino acid containing peptide will bind. Okay, clear? And I have also told you that alpha 3 domain are highly conserved. It contains a sequence that interact with CD8 cell surface molecule found on the T cell, right? So you can easily remember it, why alpha 3 domain is conserved, otherwise it cannot interact with CD8 cell surface molecule. Okay, now the 3 molecule class 1 alpha chain, beta microglobulin and peptide interact with each other solely through non-covalent interaction. And this is what I have told you about the Dowdy tumor cell. Now peptide interaction. Okay, so MHC class 1 molecule bind peptide from intracellular sources. That means all type of endogenous antigens are processed by MHC class 1 molecule. Okay, now how you can remember it, because when I have read about the immunology, there was a big confusion in my mind, and I used to forget about this, that which one will going to process which type of antigen. Okay, but now I will going to give you a mnemonic that will help you. So remember, MHC is just like our fathers. Okay, what is a first priority for our father? Our first priority for our fathers will be to handle the problems of our home, right? So MHC class 1 molecule will going to look on problems of home. Okay, that means endogenous antigen. Okay, and MHC class 1 molecule will going to look the problems of the outside. That is why exogenous antigen. Okay, I hope after that you will not forget about MHC class 1, which MHC molecule deal with which type of antigen. Okay, now, so here another important thing that I have to mention, that here on the peptide binding group, the MHC molecule will bind with this type of way. Okay, that means this entire thing is 8 to 10 amino acid long, I have told you. So the two special peptide sequence will bind over here, okay, and the middle part of the peptide will be arc away. Okay, that is very important to remember. Okay, you may get MCQ from this type. Okay, that what will be the configuration of the peptide in case of MHC class 1 molecule. So you have to write that the middle portion of the peptide will be arc away. And that is what I have written here. The anchor residue at both ends of the peptide are buried within the binding groove, right? And that is what I have mentioned here, that anchor residue at both ends of the peptide will buried within the binding groove. Okay, and that is how they hold the peptide firmly in place. Okay, and between the anchor, the part of the peptide will be arc away, okay, from the floor of the groove in the middle. Okay, so that is about MHC class 1 peptide interaction, but here I have only mentioned about the interaction. Okay, how it is processed by MHC class 1 molecule I have not mentioned till now. Okay, you will get a separate video on that particular topic. Okay, so I am now wrapping up this video here. So I hope this video will be very clear for you. So if you have enjoyed this video and if you have learned something from this video, then please support my channel. If you may also like it and share it with your friends, and please drop a comment for me as a suggestion. Okay, so bye bye.